Hello, my name's Mrs. Tyler, and today we're doing some GCSE biology revision where we're going to make a knowledge organiser. It's for triple award GCSE only, so if you're doing dual award, you don't need this content. We're looking at protein synthesis and we're looking at mutations. It's going to look something like this, lots of detail. It goes with a previous video I did looking at the basics of DNA, whereby you can stick it on and make an extra long knowledge organiser. Right, let's get started. So today we're doing protein synthesis and mutations, and this is for triple science students only. As usual, I'm going to put my title in the middle. And this time, because we've just got two topics, I'm just going to divide my page in half. So I'm going to draw a line down the middle, and we're going to start by looking at how DNA codes for proteins. And then we're going to move on to look at mutations. So we're going to draw a big picture of DNA down this side. And as you might remember from my previous video, DNA is a double-stranded spiral molecule called a double helix, and it's got a phosphate sugar backbone. So we're going to draw phosphate, sugar is a pentagon, 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 phosphate, sugar is a pentagon. How many have I done? 1, 2, 3, 4, 5. I'll do 6. Okay. So this is one backbone, and the phosphate joins to the sugar to form the backbone of one strand. Sticking out from the middle of each sugar then is a molecule that we call a base. And there are 4 types of base in DNA. One of them's A, one of them's C, one of them's G, and one of them's T. So we're just going to stick in some bases, G, G, T, C, T, A. So this is one strand of our DNA. We're now going to put in the other strand because it's double-stranded. So I'm going to draw my boxes here. The reason I'm drawing a set of boxes here is because the bases actually line up, and we call them base pairs. And they always line up in a certain way. Whenever there's a G on one side, there's a C on the other. So G, another C. Whenever there's a T on one side, there's an A on the other. So if there's a C there, there must be a G there. And if there's a T there, there must be an A there. And if there's an A there, there must be a T there. All right. So these are called base pairs. And just like there's a strong backbone holding these bases in this order, it's the same on the other side. So we've got sugars attached to each one of these. Don't worry too much if you're not drawing a very neat pentagon at all. And in between them, we've got the phosphates. Right. And then we're just going to join those up. Right. So remember we said DNA was a polymer, and the subunit of a polymer is this bit here. It's a phosphate and a sugar and a base, and the subunit was called a nucleotide. So in this section of DNA, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 nucleotides, forming 6 sets of base pairs. Let's write some of this down then. The bases pair up. A always base pairs to T, and C always base pairs to G. The reason DNA is so important is because it holds these bases in order. The order of the A's, C's, G's and T's is really important. And the reason it's really important is because it carries the code for proteins. So the order of the bases is the genetic code for proteins. So if the code is wrong, so instead of GGT it became GCT, then the protein would be wrong. All right, and that would be bad news because it probably wouldn't work. DNA then, or the bases, are read in sets of 3. And each set of 3 bases codes for a protein subunit called an amino acid. So each 3 bases codes for 1 amino acid. Amino acids are the subunits of protein. Let's have a look how this works then with these bottom 3 bases. So DNA is massive, it's huge. This could be 100,000 bases long. I just want 200 of them, and I can't send that long bit of DNA out of the nucleus, it's too big. So instead, I copy this sequence into a shorter sequence, a messenger sequence called messenger RNA. So this is copied into a shorter code. So this set of 3 would be copied. Doesn't quite work like this, but that'll do. And then the next set of 3 would be copied. And then the next set of 3 would be copied, and we get a shorter molecule called mRNA. The M stands for messenger. The mRNA then is small enough to wiggle out of the nucleus and go off into the cytoplasm to the ribosomes. Ribosomes, remember, are where protein synthesis happens. And in the ribosome, it reads the code again in sets of 3. So GAT, the bases from here, might code for amino acid number 1. So I'm just going to put number 1 there. This is the first amino acid. We can think of amino acids like a string of beads in a protein. This one might code for amino acid number 2, and the ribosome will add whatever amino acid that codes for. That's amino acid 1, that's amino acid 2. And so on and so on. The next 3 bases will code for amino acid 3, the next 3 amino acid 4, amino acid 5, amino acid 6, amino acid 7, amino acid 8, etc., etc. Until it makes a chain of amino acids. Now that chain of amino acids is the protein. And that protein is things like enzymes and hormones. So the full chain of amino acids, or the full sequence of DNA, in other words, the full gene, should make a full set of amino acids and make a protein that works. And they are things like enzymes and hormones. So this was our messenger RNA. It was a shortened copy of the DNA. So the DNA is copied to a shorter messenger, which equals mRNA. And the mRNA goes off to the ribosome, and it's the ribosome where the amino acids join together. So this happens in the ribosome, out in the cytoplasm. So the messenger takes the code to the ribosome to make the protein. So that's when DNA works, when the genetic sequence is right, the mRNA is right, and the amino acids are right, and the protein is right. Hooray! Everyone's happy. But sometimes there is a mistake in the code. Sometimes C or an A or a G are wrong, and we call that change a mutation. So a mutation is when there is a change in the bases of the DNA. This is when there is a change to the bases in DNA. In other words, the genetic code is changed. Now, mutations happen naturally. Because DNA can copy itself, the enzymes that copy it occasionally make mistakes. So we are all mutants. And the older you are, you may have noticed this, the more mutated you are. So old people will have more mutations than younger people. And that's part of the natural cycle. So they can occur naturally. As DNA replicates, it makes mistakes. Replicates. But we can increase our risk of mutations, or the chance of getting mutations, by exposure to certain chemicals and radiation. We call them mutagens or carcinogens, and the reason for that is that the mutations often give rise to cancer. So we can increase the risk of mutations by exposure to certain chemicals and radiation. We call these mutagens. They're also called carcinogens because they cause cancer. So mutations can be good, they can make things better, but most of the time a mutation that changes the protein is probably going to break the protein and it's going to cause problems. And a lot of the time that problem is cancer. Let's have a look at some of the ways this can happen then. So let's have a look when DNA is right or normal. So normal DNA. So when DNA is normal, the code should be read in sets of 3 and code for specific amino acids. So let's pretend our first code is GAT, A, G, C, and then C, C, A. This set of 3 should code for the amino acid alanine. We'll shorten it to A. This set of 3 should code for the amino acid serine. We'll call it S. And this set of 3 should code for the amino acid proline. We'll call it P. And these should join together like a string of beads and make part of a protein. Let's pretend that these amino acids are making an enzyme. Now let's look then at what happens when the mutation occurs and this code is changed. And at GCSE Triple, there are 3 types of mutation or 3 types of change you need to know about. So we're going to draw 3 arrows down from here. So 1 to just here, 1 quite a long way down, and 1 to just there. Right, so 3 types of change. The first change then is called a substitution. As the name suggests, it's when one of the bases is substituted or swapped for another one. When a base is swapped for another. Sometimes the swap will change the amino acid, so we'll change the protein. But the thing about substitutions is sometimes the swap won't change the amino acid, so won't change the protein. So sometimes this doesn't alter the protein. So substitutions can have no effect at all. Let's have a look how this works. We're going to take our normal healthy DNA code, GAT, AGC, CCA, and we're going to substitute this C for a T. So the first 3 are going to be the same, GAT. The next 3, instead of being AGC, they're going to be AGT. And that T is the substitution. We've swapped the C for a T. The next ones are the same, CCA. These codes then code for the amino acids. So GAT coded for alanine, amino acid A, and AGT still codes for serine, S. And CCA is the same, so it still codes for proline. So this string of amino acids is still the same. Which is good, because it means the enzyme is going to be still the same shape, and it's going to work. The active site will still work. Another type of mutation then is called an insertion. And we'll do about this down here. Let's draw a box around it so it stands out. Insertions, as the name suggests, is when extra bases are added or inserted. This can have a very damaging effect because inserting an extra base can change not just one amino acid, but all of the amino acids. So this can change one amino acid or all of them. And if this happens, that's obviously bad news. Let's go back then to our sequence, GAT. This time we're going to add an extra T in here. So we've got GAT at the start, but instead of AGC, we're sticking a T in, so it becomes TAG. And what happens is that C gets knocked on to the start of the next one. Okay? So that becomes CCC. And then that A gets knocked onto the next one, etc., all the way down the rest of the amino acid. So in this insertion here, that one's being added. This one starts off the same, still codes for amino acid A, but this time TAG is wrong. It codes for tyrosine, amino acid T, not amino acid S, so it's wrong. CCA codes for proline, but now we've got CCC, which might code for a different amino acid. I'm going to call it M, which is also wrong. And the next one will be wrong. And the next one will be wrong. So all of these are wrong. And what that means is the protein chain probably isn't going to make the right shaped enzyme. It's probably going to have a funny shaped active site, and it's not going to work. All right. So insertion mutations, adding one extra base or more than one, can cause a knock-on effect down the rest of the chain, and that can have a really damaging change to the protein. The last type we're going to look at then is called a deletion mutation. And again, thankfully it's got a sensible name. Deletion means something's been deleted or removed. So in this case, bases or a base have been removed. And again, just like an insertion, it can just affect one amino acid if the code stays the same, or more likely it will affect all the amino acids. So if you remove 3 bases, then you just knock out one amino acid and all the rest are the same. But if you remove any other number than 3, all of the amino acids will be changed. So once again, we're going to go back to our original sequence. GAT is our starting triplet, our starting set of 3. We're not going to mutate that one, but this time we're going to delete. Okay, we're going to remove a base. So we're going to pretend that we are deleting that A. So the A is deleted, which means we get GCC for the next one. And then we get CA whatever. So amino acid A is the same, but this time amino acid S is now amino acid L, and this one, instead of being amino acid P, might be amino acid T. And they are wrong. And the rest of them will be wrong. And again, that's bad news because it means if this is coding for an enzyme, then the enzyme is probably not going to have the right amino acids and it's not going to work. It's not going to be the right shape. We say it's not going to be functional. And that's it. Well done. So there's a lot of detail on triple, on how DNA works coding for proteins, and then what happens when that code goes wrong. You know the routine. Your job now is to go away, look at this, test yourself on it, and have a go at some exam questions. Well done, everybody. I'll see you next time.